Right guys, so we're looking at the specific immune response, which is the immune response for specific antigens that are parts of pathogens. And usually an antigen is represented by a few amino acids that are foreign and non-self. And so we're going to, in this presentation, look at the various characters involved in the immune system. So first of all, here we've got a macrophage. A macrophage is a phagocyte, and a macrophage will engulf and digest through phagocytosis a pathogen, and after digestion a macrophage will present the antigen on its cell surface membrane, okay, and therefore we call it a antigen presenting cell. The next character in our story is the T helper cell. The T helper cell is going to be the intermediary within the immune system, and it contains, the most important thing about them is it contains a T cell receptor, and a T cell receptor is going to interact with a macrophage later or a B cell, and T helper cells also release cytokines, and cytokines are chemicals of the immune system, and they are going to affect either T cells themselves or other cells. Okay, a B cell, also called a B lymphocyte, carries membrane-bound antibodies, and each particular B cell has a particular type of antibody. And as I said in the lesson today, there are millions and millions of types of antibodies, and therefore millions and millions of types of B cell. And a B cell also has the ability to, when it recognizes a pathogen through its antigen and the antibody binds to it, it engulfs and digests that pathogen and then also presents on its cell surface membrane the antigen to the rest of the immune system. So that's a B cell. Okay. The last actor in this play is a killer T cell, and a killer T cell is going to be involved in the killing of viruses later. Okay, so if we move forwards. Okay, so I talked today in the lesson about a macrophage being the first port of call for a pathogen, digested and presented on what we call Mhc protein. And so on that macrophage there, the purple cell, you can see that there's a purple rectangle. That's Mhc protein. And Mhc protein, like a pair of hands, holds the antigen out to the immune system. And so you can see the red triangle there is the antigen. Now, there are millions and millions and millions of types of T helper cells. And T helper cells, as I said earlier, have a T cell receptor. And that T cell receptor will be individual to that T helper cell. So you can see there's three types of T helper cell, although there'd be hundreds of millions. You can see one with a semicircle, one with a rectangle, and one with a triangle. And the T cell receptor on the T helper cell will recognize both the antigen and the Mhc protein holding it. So it recognizes the whole complex, okay? And so when, by chance, the T helper cell comes across the macrophage, we can see what happens. The correct T helper cell is going to bind to the Mhc protein holding the antigen, okay? And that is going to cause the T cell to stimulate itself, okay, with cytokines. And it releases the cytokines into the immediate area, and this causes that particular T cell with that particular T cell receptor to proliferate. So we've now got more of the right type of T cell with T cell receptor out there, okay? And you can see all of these cytokines are the things that cause the T cell to do that, okay? So what do we have so far? Well, we've got a macrophage, which has presented antigen. We've got a T cell, which has recognized the antigen through its T cell receptor. And when it's bound, it's caused itself to proliferate. So we've now got loads of T helper cells with the right receptor. Now, meanwhile, elsewhere in the immune system, and this is often in the lymph system and lymph nodes, B cells, the B lymphocytes with the particular antibodies which are membrane bound, and you can see on the screen there's three different types. They are hanging around waiting for their complementary pathogen with the antigen to bind with them. And they might wait forever and it might never happen, and one day it might happen. And so when that happens, like the macrophage, B cells are antigen presenting cells. They will take the pathogen in and they will digest it and present it on their cell surface membrane. So let's see what that looks like. So there we've got our three different B cells, okay, and we've got some pathogens, and that particular B cell is the one that is complementary. It, by endocytosis, takes in the pathogen, digests it into its constituent amino acids, and then presents on the surface of the cell, once again with MHC2, presents the antigen to the immune system. And so we've got T cells floating around because they divided with the right T cell receptor for that antigen in MHC. And so we're hoping, I hope you can see ahead, that those T cells are going to come across one of the B cells. And lo and behold, this is what happens. So we can see on the screen there, we've got a B cell presenting in MHC2, the antigen, and we've got the correct T cell with the correct complementary T cell receptor. And what happens? Well, the T cell receptor is going to bind with the MHC and the antigen and release cytokines. These cytokines are going to stimulate the B cell. You're the one that we need loads of because you carry the right antibody for the pathogen. And so we get what is called, you can see here, it's clonal selection of that B cell. Okay, you are the right one. Okay. So what can we see on the screen? Well, we can see that one B cell has turned into 9, is it? And therefore there's been expansion. So this is clonal expansion of the correct B cell. So these B cells are proliferating massive amounts of mitosis, and they turn into B effector cells. And these then are able to turn into plasma cells or produce plasma cells. And plasma cells are very specialized cells because they release soluble antibodies which are not membrane bound into the body systems, into the lymph, into the blood. And they are, and like I described, sugar being dissolved in tea, there's never a sip you take which doesn't taste of sugar because it's dissolved everywhere. The soluble antibodies, hundreds and hundreds of millions of that right type, that right shape of antibody floods the body systems and is going to bind with the pathogen. There's nowhere for the pathogen to hide. Now, the pathogens with which are bound to antibodies are, this isn't shown in this particular sequence, but we'll talk about this later. They are not killed by the antibodies. But what happens is the antibodies bind to them, and through a number of mechanisms, they cause the pathogen to be either eliminated or neutralized. So, for example, neutralization by antibodies is where the antibodies bind to the important receptors and membrane-bound parts of the pathogen, which prevent it from doing what the cellular processes that it would need to do. So it's a bit like it's ineffective. Okay, you can actually see that antibodies have two, like they're Y-shaped, so they've actually got two heads. It means that antibodies can bind more than one pathogen at any one time, and so they're able to stick clumps of pathogens together. We call this agglutination. Okay, the other thing that antibodies are able to do is they're able to, by binding to the pathogen, they are able to, with their tails of the Y, they are able to dock in with phagocytes. And we call this opsonization because it means that you have receptor-mediated endocytosis, where instead of phagocytes coming along and just randomly coming across them and phagocytosing, there's like a docking mechanism of the tail of the antibody and the membrane of the phagocyte. Okay, so in summary, what antibodies do is they either neutralize or they make the pathogen a sitting duck for macrophages and phagocytes to come along and, like sharks, just eat them. When I say eat, obviously I mean engulf, digest, and it makes it easy for the immune system to kill them. At the same time as this, when the B cell was proliferating and producing effector cells and plasma cells, B memory cells are formed, and the proliferation of the T cells earlier forms T helper memory cells. And we talked about this at GCSE, but it means that there's an immunological memory of that pathogen, which means that we can have a faster and greater response in the future if there is a reinfection. So that is what we call the humoral response. It takes place in the fluids of the body. In the olden days, Greek medicine used to say the humours were the fluids. And so the war takes place in the lymph, the blood, and the fluids of the body. And so in summary, and I hope you can visualise this, and if you want to get a bit of paper and just jot this down, we have a macrophage which meets a pathogen. It engulfs and digests it and presents antigen on its cell surface membrane because it's an antigen presenting cell. It presents the antigen on Mhc protein. A T helper cell with a complementary T cell receptor to that macrophage's Mhc2 and antigen will bind to the macrophage, and that will cause activation of that T helper cell by the release of cytokines. The T helper cell will replicate itself, and because it's the right one, and there will therefore be loads of T helper cells moving around the body. Meanwhile, elsewhere, a B lymphocyte with membrane-bound antibodies will have also met another pathogen with exactly the same amino acid sequence and antigens, and it will have bound that pathogen and digested it and then presented antigen on its cell surface membrane with Mhc protein. The T helper cells, one of the T helper cells, the many we now have, will bind with the B lymphocyte Mhc2 holding antigen, and that will cause the release of cytokines by the T helper cell, which will cause clonal expansion of the B cells. You're the right one, we need you to produce antibodies, go, 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 go. Massive amounts of mitosis, massive amounts of B cells produced. This produces B effector cells, which produce plasma cells, and it's the plasma cells, the B plasma cells, which release soluble antibody, which flood the fluids of the body. There's nowhere to hide for the pathogen in that case. The antibodies bind to the pathogen and they either neutralize it, they agglutinate them, or there is optimization and phagocytes come along and engulf and digest all of the pathogens. And that is the humoral specific immune response.